Okay, let's see if my microphone will stay connected for more than 10 seconds. This will be our chapter that is a very brief review of some chemical principles, because in chapter 3 we're going to look at putting all of those chemicals together to build our biological molecules. You've probably seen these ball and stick models in your chemistry class, where each ball represents an atom, and that ball has the number of spots on it to where that atom can make connections to other atoms. And this tells you which ball color represent which atoms. It can change from kit to kit. And we're not going to be building ball and stick models, but you will see images of this with our different molecules. So as our little brief reminder, the nucleus of an atom contains the neutrons, which have no charge, and the protons, each which have a single positive charge. And then there are the shells, or the orbits, for the electrons, each of which has a negative charge. And for a neutral element, it is defined by its number of protons, and that does not change. The number of electrons is typically the same as the number of protons, because an element has no charge. If there's a charge on it, that is an ion, and then that we will talk about later. The atomic number is the number of protons. The mass number is the sum of the number of protons and neutrons. And the electrons do contribute to mass, but not significantly. So in this class, we're going to stick with the atomic number being a whole number that is the sum of the number of protons and neutrons. So if we look at our periodic tables, the chemical symbol is the abbreviation, the atomic number on the upper left, and the mass number below it. Both of these are carbon. Both of them have the atomic number of 6, because that does not change for an element. The mass changes means that the number of neutrons is different, and this means it is an isotope. So a similar type. It's still carbon, but the mass number is changed because of the number of different neutrons. You may have heard of carbon-14 dating. That is used, one of the elements used for dating old fossils, and that means it would have yet one more additional neutron. So carbon dating, about 50,000 years old. If it's much older than that, carbon dating can be used as a starting point, but other isotopes of other elements that are more accurate are used. The periodic table, I'm sure you're familiar with this. There is one in your free textbook so that you can enlarge that and look at it as you need. But again, it shows you where the atomic number is, the symbol, and the atomic mass. And the periods color-coded by what types and what period they fall into. In a biology class, we're going to be fairly limited in the atoms that we need to know about. Carbon, of course, because we are carbon-based life forms here. Also oxygen. Oxygen is critical for us to breathe. It's not so critical for other organisms, but it is a very important atom in organic molecules. Nitrogen as well, hydrogen, and we're also going to need phosphorus and sulfur. Other elements are very important, like sodium, magnesium, potassium, calcium. Those all form ions and are critical in functioning of our cells, particularly our nervous cells. So the Bohr's model was developed in 1913. So the electrons are in their shells. In your chemistry class, you learned about s orbitals and p orbitals, and we're not going to go into that much detail, but we are going to talk about those electron shells, where typically that first shell has a maximum of 2 electrons, and the next shell has a maximum of 8 electrons, and so on. So the higher the atomic number, the higher the number of protons, And as we exceed the number that fit in each shell, we get an additional shell that will be the valence electrons that determine how that atom can interact with other atoms. So there's period 1, and we've got the first shell for hydrogen just has 1 electron. The atomic number of hydrogen is 1, so it has 1 proton. Because it has 1 proton, it also has 1 electron, and because its atomic number is 1, there are no neutrons. You don't have to have neutrons. They contribute to mass, but they are not necessary for the functioning of the atom. Helium has an atomic number of 2 protons, and it has 2 electrons. And we see in this group 18, in period 1, we fill that inner shell. Hydrogen has the capacity to bond with one other atom because it would like to have another electron to fill its shell. A filled shell is more stable. Helium, filled shell. In period 2, as we go through, we start out with 1, now we have 4 in group 14, we have 2, 4, 6, 7 outer electrons in group 17. Get to group 18, 2, 4, 6, 8. Neon is filled. We get to period 3, we're filling the third shell, and again as we go across we get more and more electrons. That group 18 have that full valence shell, and that means these are our noble gases. They don't bind to anything else because they don't have any room in their valence shells to bind to any other atoms. So in group 1 we have room for 1 electron for hydrogen. Lithium has 2 in its inner shell but only 1 in its outer shell, so it either needs to bind to 7 things or it's more likely to become an ion and give up its 1 electron to another element. In group 14 we've got 4 valence electrons for both carbon in period 2 and silicon in period 3. That means it can bind to 4 things to fill up its shell, and that's why in science fiction you often see them hypothesizing about silicone-based life forms, because its valence shell is the same as carbon, but because it has an additional orbit, it's larger and it is less stable. Fluorine and chlorine have 7 valence electrons. They easily bind to one other element, but they are often become ions by accepting electrons from another element. For instance, sodium has 1 valence electron, chlorine has 7. Sodium ion easily forms when this electron is released, and chlorine becomes an ion when it receives it. We get an ionic bond and we get sodium chloride. So when we start talking about bonding, we will see the different types of bond. Ionic bond is one sort. So there's an actual pictures of our orbitals. How water forms. Oxygen has an inner orbit with 2 electrons. Its outer orbit has 2, 4, 6. These 2 aren't shown as next to each other because they will be circulating, and it more easily depicts how bonds can be formed. Hydrogens each have the capacity to form 1 bond, so this hydrogen can bond and this pair of electrons will be shared. Hydrogen can bond here to share a pair, and we end up with H2O. So these 2 electrons, one that was originally circulating around oxygen and one around hydrogen, and likewise the other pair, are now equally shared. So either the white or the red one could be circulating around either nucleus. We have 2 pairs, unshared electrons on oxygen, and that is why water gets shown as a bent molecule. These extra unshared electrons will be trying to get away from each other because they have the same charge. They exert electromagnetic force on the hydrogens and push them to the sides. So when we bind 2 oxygen atoms together, we actually create a double bond. So each covalent bond, co meaning together and valent, it's our valence electrons, is 1 shared pair of electrons. So in an oxygen molecule and others, if we have a double bond, we have two shared pairs of electrons. Single bond indicated by the single solid line between atoms, one shared pair of electrons. Double bond indicated by two solid lines, we have two pairs of electrons. So covalent bonds, one bond is a shared pair of electrons. And here's a better depiction of our sodium chloride ionic bond. Sodium has that one lone electron in its outer shell. Chlorine has 7. If sodium donates its electron, now sodium does not have an electron in that shell. Its shell below that is full, and chlorine now has a full valence shell of electrons. They are not sharing electrons. Sodium no longer contains that electron. Chlorine is in full possession of it, and now the sodium has a +1 charge because it lost one of its negatives, and chlorine has a -1 charge because it gained a negative charge. So when we look about how these covalent bonds affect a molecule, we can have polar and non-polar covalent bonds. Water is polar and covalent. Covalent because we are sharing electrons. It's polar because the charge distribution across the molecule is uneven. These little squiggles are lowercase Greek delta. And so we have delta means partial. We have a partial positive charge on each hydrogen. We have a partial negative charge on the oxygen. And those electrons again are pushing on the hydrogens and it becomes bent. Methane gas is covalent. We have solid lines drawn between the atoms, but it's non-polar. Each of the atoms are equally sharing the electrons around their nucleus. And then we have carbon dioxide is polar and covalent, but it is not bent. Water is bent. The carbon dioxide is not, because the polar bonds are on either side of the carbon. They're pushing on each other equally, and you get a linear molecule. So that polarity is very important into how water acts in living systems. So this is a microscopic image of oil and water. The oil is clinging to the oil, the water to the water. So we form these droplets of the oil. We could get them to mix and visibly look as if they're evenly distributed, but they're not going to combine because the water is polar and the oil is non-polar. And another way to think about that is hydrophilic, being water-loving. So water is obviously hydrophilic, but anything that would dissolve and mix well in water is also going to be polar. It will be water-loving, hydrophilic. And things that do not dissolve in water do not like water. They are hydrophobic. So hydrogen bonding is neither shared electrons, like a covalent bond, nor are electrons donated and received. It is because of the partial positives on each of the hydrogens and the partial negatives on each of the oxygens that we have sort of an electrostatic attraction between them. In liquid water, the bonds, the hydrogen bonds, are constantly breaking and reforming. But when we have ice, the solid form of water, then those hydrogen bonds are literally frozen, and it makes a lattice. That lattice means that there's tiny bits of air spaces in between, and that is why ice floats. It is the only substance where the solid form is actually less dense than the liquid form. Now when we look at how sodium chloride, table salt, reacts in water, the reason why it dissolves so well, unless you put an enormous amount in, is that each negative chlorine ion attracts the partially positive hydrogen atoms on the water molecule. And it becomes surrounded by those water molecules, and now we're not going to get the sodium ions attracted to it. Each of the sodium ions is positive, and it attracts the partially negative oxygen portion of each water molecule, and it gets coated and won't recombine with the salt. And those are called the spheres of hydration, because we form a sphere, we soak the water around the ions, and then they don't bind. Another property of water is its cohesiveness. We have those hydrogen bonds connecting the water molecules, and we have the weight that's pulling the needle downward due to gravity, but the surface tension is pulling upwards. Now, of course, if we put the pointy side down, it's going to sink. We'd have to place it very gently on the surface for that to happen. But surface tension allows this sort of thing, or for insects to be able to walk on water. It's also what allows us, if we're very gentle, to slightly overfill, especially a very narrow tube like a test tube or a graduated cylinder. That round bubble on the top is due to the surface tension and the hydrogen bonds between neighboring water molecules. Capillary action is when you put an empty tube, the adhesive forces by the internal surface of the glass are exceeding those cohesive forces between the water molecules, and they get drawn up. It's not going to go all the way to the top, but it does go part way up the tube. In our face-to-face labs, we use capillary tubes to draw up small amounts of liquids and apply them to surfaces or into test tubes. If you put a straw into your smoothie or your coffee or other liquid, you get some capillary action where the liquid gets drawn up into it. There's a water strider insect also taking advantage of those cohesive properties and surface tension of water. pH stands for potential hydrogen ions in solution. And it does have to be in solution. You can't take the pH of the table. You can't take the pH of solid things unless they are ground up finely and put into solution. And pure water is a pH of 7. And anything below that is acidic. Anything above that is alkaline. And so urine should be somewhat acidic. Coffee, tomato juice, all the way down to your stomach acid, and things that are alkaline, seawater, baking soda. What isn't on here? Blood. Your blood is a pH of about 7.4. It is not neutral. And it has to do with calculating the concentration of hydrogen ions. So a low concentration of hydrogen ions, the pH is calculated by taking the negative log of that concentration of hydrogen ions. And so that's how we get a higher number with the alkaline when there's very low amounts of hydrogen ions, and a low number when there is a high amount of hydrogen ions. And as a reminder, each pH unit change is a 10-fold change in hydrogen ion concentration. So the difference between distilled water and seawater, it's 1 pH point, but there are 10-fold fewer hydrogen ions in seawater than in distilled water. Likewise, between 7 of water and 5 of coffee, that's 2 units. That means coffee has 100-fold more hydrogen ions than does distilled water. So when we look at our bodies, right? You might see things about alkaline water and alkalizing your system. It's really not possible. Our bodies function in a fairly narrow pH range, and we have buffers. If you survived your Chem 1A, which you had to get into this class, you did titration experiments, and you see where that point changes. Well, buffers are chemicals, whether in a test tube or in our bloodstream or inside of our cells, that prevent dramatic changes in pH. They either accept hydrogen ions to make it less acidic, or they donate hydrogen ions to make it less alkaline. So ingesting alkaline water, yeah, it might temporarily change the pH in your stomach, but not for long. The gastric acids there would neutralize that. And by the time it gets into your bloodstream, the buffers will be preventing any sort of dramatic pH changes. So the blue arrow shows raising pH. We make carbon dioxide. So carbonic acid would be if we have hydrogen and carbon dioxide combining. We can make that as a proton donor, an acid. There's the proton, there's the base. But that gets formed when we breathe out as water and carbon dioxide. So when we get to metabolism, we'll see that the foods we eat, by the time we metabolize them fully, we get energy stored from that, and we release the waste products either as urine or feces, but the chemical waste products are in our breath, the carbon dioxide and the water. So there's methane. We are not going to calculate bond angles. That's what you did in chemistry. But we're going to look at how those polar and nonpolar bonds affect biological molecules. Methane, not a biological molecule in terms of building in us, but it is a biological molecule in terms of waste. It is the gas that is released when flatulence occurs. So that is farts. There's a carbon molecule, 4 evenly spaced hydrogens. So there's methane, which is CH4, and there'd be only one way those 4 hydrogens could attach. Those are single bonds, so 4 separate shared pairs of electrons, and all of the atoms share the electrons evenly. Ethane, the ETH, when we're looking at organic molecules, means it's 2 carbons. So does acete, ACET. So the 2 carbons are bound to each other. Each carbon then finishes its shells by attaching to 3 hydrogens. And we can have a double bond. ANE tells you the carbons have a single bond. ENE tells you they have a double bond. You can have a triple bond between carbons, and that is YNE, it'd be ethine. Circular formations certainly happen in biological organic molecules. So pentane, ANE, all of the bonds between the carbons are single. Pent, P-E-N-T, stands for 5, like a pentagon, and that is the way the carbons form. And it's cyclo because it has made a circle. So once the carbons are bound, we finish off the 4 bonds each carbon can have by putting hydrogens on it. Likewise, cyclohexane, ANE, it's single bonds. Hex, it's 6. Cyclo, it formed a circle. And now we get into different types of terminology that you just have to memorize. Benzene. ENE tells you there are double bonds. You just have to remember they alternate. Double, single, and then each carbon has only 1 hydrogen because on one side 2 bonds are to another carbon, on the other side 1 to another carbon. That takes up 3 bonds. There's only 1 left. And then lastly, we show an example where nitrogen can be incorporated into the carbon circle. We have pyridine. The nitrogen into the circle of carbons becomes important in DNA. Our DNA molecules all have nitrogen incorporated into the circular carbon molecules. So just like we have isotopes with our atoms, we have isomers with our molecules. So both of these have 4 carbons. This is butane, all single bonds. This is isobutane. It still has 1, 2, 3, 4 carbons. It still has the same number of hydrogens, but we have moved one of the carbons into a branch point. So that's called a structural isomer. A geometric isomer, they are arranged differently around a double bond. And so this is butene. We still have 4 carbons, but between 2 of them, we have a double bond. So we're going to have fewer hydrogens. And we can see the difference is in this instance, in the upper right on the cis butene, we have a hydrogen, and on the trans-butene, we have a methyl, a CH3. This will become important when we're talking about fats. You've probably heard of trans fats. And trans fats mean we have functional groups on the opposite sides of the double bond, and cis fats, we will have them on the same side of the double bond. And that does make a difference. It looks, when it's drawn 2D, like, so what? We have the same number of atoms, the same formula, but the arrangement, particularly in biological systems, means that enzymes won't be able to recognize and metabolize when that atomic arrangement is different. And I never can say this word. Enantiomers are mirror images. You cannot lay them over each other. So it would look like, well, you just have a carbon with what, a hydrogen, a chlorine, a bromine, and a fluorine. Why isn't that the same? Well, it's like our hands, right? If I do this, they lay over each other. But because they're mirror images, if I have both my palms facing out, I can't put one over the other and make it the same. That's what the mirror image is for the enantiomers. So this is looking at the fats. This oleic acid is a cis fat, and oleic acid is a trans fat. So when the hydrogens are on the same side of the double bond, it's linear. And when they're on the opposite sides of the double bond, it gets bent. So when you have, the bend is caused by the cis formation. My bad, I read it in the wrong order. So the cis configuration is the bend, because when you have the functional groups on the same side of the molecule, that's going to cause a bend. When you put them across, it's what causes the linear. So the trans fats become more linear, and that makes it look more like a saturated fat. The enantiomers are the mirror images. Alanine is an amino acid, and only the L shape is the one that's used to make proteins. So when we get into our chapters about protein formations, all of the amino acids we talk about will be the L formation, even though D formations can exist. Functional groups are an arrangement of atoms that when you attach them to a larger molecule, that is the reactive portion. It's what gives the molecule its function. So the structures are listed here. The R means the rest of the molecule. So the name of hydroxyl, we have an OH. It is polar and hard to tell from this structure without seeing the Fisher the actual atomic interactions with their valence electrons, but OH is 2/3 of a water molecule. So whenever you see lots of hydroxyls, you know it's going to be polar. Methyl, CH3, we saw that with methane, that is nonpolar. All of the carbon is completely coated in hydrogens. Carbonyl, we have rest of the molecule on either side, and there is a carbon with a double bond to an oxygen, and that is always going to be polar. We're going to have some unpaired electrons on that oxygen. A carboxyl is kind of like a carbonyl, where one of the R's is replaced by a hydroxyl. So we have the carbonyl carbon, but you also have the OH. So this whole thing is the carboxyl. It is charged because in solution it tends to release a proton, and that makes it an acid, because acids are proton donors. The amino is the nitrogen with 2 hydrogens. It also becomes charged because it accepts hydrogens. So chemicals that accept protons are bases. So acids donate protons, bases accept protons. Phosphates will be very important, very complicated. We have a phosphorus. It is double bound to an oxygen. It's got 2 hydroxyls, so it's going to be polar. It's also charged because it releases hydrogen ions from its hydroxyl groups, and because it does that, it is acidic. And then sulfhydryl is also polar. It's got a sulfur and a hydrogen, and sulfur is directly below oxygen on the periodic table. So when you see an SH, it's going to have similar values in terms of polarity as an OH. So this is a ball and stick model of some of the components of DNA. And we have thymine is one of the nitrogenous bases. So there are nitrogens in our carbon ring. And this represents one of the strands of the DNA. This is the other strand, and we get hydrogen bonds forming between our bases. And that's what's going to form a spiral staircase. So the individual components to build one strand of DNA are covalently bound to each other, but then the 2 separate strands of DNA will have hydrogen bonds between them. So that is a super quick run through of some chemistry. That will wrap up the first week's videos. So you will have until noon on Wednesday, August 26th to post your initial thoughtful question or comment. That will open up the board to see others' responses. And then by midnight, or actually 11:59 p.m. on August 26th, you need to reply to at least 2 of your classmates' comments. And today for me is Monday. So each week, the day after you have done your lecture assignments, I go in and read through all the comments and assign your grades. Likewise, the day after your lab. Your labs are due on the Thursday of that week. And so the first lab will be due on Thursday, was that the 27th, 24th, 25th, 26th, 27th? Yes. And also by 11:59 p.m. So people that have been having trouble with the submission, we are troubleshooting that. We will get it done. Things will smooth out. And as always, there's a chat in your Canvas shell. So any comments, things you've discovered, troubleshooting solutions or problems, put them there so the whole class can see them and we can help each other out as much as possible. So far, so good. People are getting in and getting stuff done. I appreciate all the effort. It's going to be a bumpy semester, but we will help each other through. See you next time.